There's still some people trickling in right now, but I guess we will start because it's 8 o'clock. I welcome you to our second short talk session of this symposium, which is on GPCR signaling and signaling partners. And our speakers today will be Joy Lee from the University of California, San Francisco, Ben Clements from the University of Michigan, Amod Godbole from the University Hospital in Jena, and Michael Wedemeyer from the University of Texas Health San Antonio. I apologize, the order of speakers is going to be a bit different than what is displayed here, but this won't make the short talks any less exciting. So I'm going to give the floor directly to Amod. If you want to start and share your screen, the floor is yours.
Okay. Okay, so I hope you can see my presentation.
Yes.
Okay, so I would like to thank the organizers for organizing this conference, as well as to invite me to show part of my data. So my name is Amod Godbole. I'm a postdoc in the group of Carsten Hoffman in University of Jena. And I would like to show some data about how adenosine A1 signaling gets regulated by GRKs and arrestin. So adenosine is a purine. This is how it looks like, and it can be produced inside the cell or outside the cell by breakdown of ATP via AMP into adenosine. And adenosine is also the natural agonist for adenosine receptors. So there are A2A and A2B receptors, which are GS coupled, and A1 and A3, which are GI coupled. They have tissue-specific differences in expression and signaling, but the overall effect of their signaling is homeostasis. So what happens in situations like cell damage or hypoxia is that that there is an increased level of ATP outside the cell, which means there is more adenosine. And this leads to more signaling and cyclic AMP regulation. And this has been shown to be connected to more pro-inflammatory cytokines, which leads to more inflammation, sepsis, tumor, and COPD. The level of adenosine itself can be controlled by degradation into inosine by deaminases, as well as by transport of adenosine through transporters inside the cell. But the mechanisms of how the signaling of the receptor itself is regulated from inside the cell, this is not clearly understood, because there are some immune deficiency diseases where the transport or the degradation doesn't really work. So this is very crucial to understand how signaling of these receptors is actually regulated from inside the cell. The adenosine A1 has been quite tough to actually understand how it gets regulated. So the main questions were to understand how adenosine A1 gets phosphorylated by which GRKs, for example, as well as which beta-arrestin is responsible for interacting with the receptor and in which fashion. And then also to understand if this can bring down the receptor expression by internalization, and how all of this actually influences cyclic AMP regulation. So we first started with some simple experiments where we just put the adenosine receptor CFP in HEX cells, and we saw that it's on the membrane. And after stimulation with adenosine, this is a natural agonist. Half an hour later, this receptor was internalized into endosomal compartments. We also tried this with other agonists, for example, NECA, which is a non-specific adenosine agonist, as well as CPA, which is an A1 specific agonist. And we saw that all these agonists are able to induce internalization compared to the antagonist DPCPX. The next question was what is the root of this internalization, because there are some reports for the A2A receptor which shows that it uses caveolate instead of clathrin to internalize. So we also looked if A1 also internalizes via caveolate. For this we used methyl beta cyclodestrine. This is an inhibitor of caveolate by depleting the cholesterol, and as you can see, in the presence of MBCD, the internalization is severely inhibited. To see the next step of internalization, we wanted to see if the pit of caveolae, if this is excised by dynamin, and for this we use dinosaur, which is an inhibitor of dynamin. And here you can see in the absence of dynamin, the receptor is internalized into endosomal compartments, whereas in the presence of dinosaur, the receptor remains on the membrane. So this showed us that this receptor internalizes via a caveolae and a dynamin-mediated route. The next step was to see if beta-arrestines are actually helping this internalization, and especially which beta-arrestin. For this, we used beta-arrestin knockout cells from ASCA Inu, and we transfected these cells with the A1 receptor CFP in green, and then rescued them with beta-arrestin 1 or beta-arrestin 2 in magenta. Beta-arrestin 1 was not able to induce any internalization of the receptor, which also corroborates with some older data. And if you rescue the cells with beta-arrestin 2, we could clearly see an internalization of the receptor in these compartments, as also shown in this quantification. Sorry. Then we had a closer look actually at the image sequences from this microscopy data. And in the green channel, you will see the receptor. So after stimulation with adenosine, you can see that there are clusters of adenosine receptor in these compartments. But if you look at the channel of the beta-arrestin, the arrestin translocates to the membrane, but it remains on the membrane as clusters, and it actually does not go inside with the receptor. So we wanted to quantify this in a better functional way, and for this we used a bystander breath assay in which we tagged the receptor or the beta-arrestin with nanoluc as a breath donor, and we tagged the 5V as an early endosome marker with neon green as a breath acceptor, and then we tracked the internalization of the receptor or the arrestin. In this breath change over time, you can see that only the receptor, which is shown here in blue, internalizes into early endosome, whereas the arrestin does not go into endosomes. It more or less sticks on the plasma membrane. This is also pretty clear that beta-arrestins get or interact with receptors which mostly tend to be phosphorylated by GRKs. And to understand which GRKs are actually phosphorylating this receptor, we used our GRK knockout cell lines, and we transfected these cells with the A1 receptor nanoluc as a breath donor, and beta-arrestin 1 or 2 halo as a breath acceptor, and then individual GRKs 2, 3, 5, or 6 to understand which GRK phosphorylation mediates this interaction. And then we plotted the breath change over time. Needless to say, the beta-arrestin 1 did not interact with the receptor. However, beta-arrestin 2 interacted with the receptor only in the presence of phosphorylation mediated by the cytosolic GRK2 and the membrane-anchored GRK6. So this was pretty clear that GRKs are essential for beta-arrestin interaction with the receptor. Then we wanted to understand which serines and threonines are actually phosphorylated by the receptor, because as we know, GRKs are serine threonine kinases, and we had a look at the serine threonines of the A1 receptor. There are only 3 serines in the third loop of the receptor. Yeah, so this is the SASS sequence here in the third loop. To understand if these serines are responsible for the phosphorylation, we mutated these serines into alanines, and we called it as the SAS mutant. And we investigated if these are essential for the interaction. So the wild type receptors, obviously, they interacted with the arrestin in the presence of GRK2 and 6. However, when you replace the serines with alanine, this interaction is completely lost, showing that these serines are essential. The next step, or when we were looking at this, at the number of serines, we were a bit puzzled because we saw that it's very low amount of serines that we see on this receptor, yet it requires the arrestin to go to a phosphorylated receptor. So we took some lessons from the paper of John Janetsko last year. where he classified the GPCRs into two classes. Yeah? One class forces the arrestin to be dependent on PIP2 as an anchor, and the other class which does not force the arrestin to be dependent on PIP2 as an anchor. And we thought that maybe this is maybe how the adenosine A1 also works with beta-arrestin 2. So for this, we use a beta-arrestin 2 mutant, which he described, which is deficient in binding to PIP2. And then we wanted to see if this anyhow affects the recruitment and internalization. So we went back to the beta-arrestin knockout cells. We expressed the receptor in CFP and the beta-arrestin 2, either wild type or the PIP2 binding deficient mutant. So in the wild type, you can see in magenta, the beta-arrestin goes to the membrane after stimulation and it sticks there. That's also from the previous data. But if you use a beta-arrestin 2, which is deficient in binding to PIP2, it does not go to the membrane at all, so there is no recruitment to the receptor or to the membrane. And we confirmed it further if this loss of PIP2 also affects the function of beta-arrestin by looking again at the internalization of the receptor. So if you use a normal wild-type arrestin, the receptor internalizes into early endosomes, as you can see in this red curve. But if you use a PIP2 binding mutant, the receptor does not also internalize. So this shows that the adenosine A1 forces the beta-arrestin 2 to use PIP2 as an anchor to recruit to the membrane, as well as to do its function of receptor internalization. To really understand how does phosphorylation and arrestin actually influence cyclic AMP production, we went back to the EPOC sensor. So as you saw from the movies, the receptor and arrestin are not really sticking to each other. The interaction is super transient. So we really wanted to see if this can really regulate cyclic AMP signaling of the receptor. And for this, we used the EPOC 187 sensor from KCI from Netherlands to look at cyclic AMP. For this, we used two types of HEC cells. These are HEC 293 cells, which are normal. They have all GRKs and all arrestins in the endogenous level. And we compared them to the GRK knockout cells, in which the GRKs are knocked out, so there is no phosphorylation and also there is no arrestin binding. So in the normal HEC cells, we see a pretty slow cyclic AMP inhibition, which is fine. But if you remove the GRK mediated phosphorylation as well as arrestin recruitment, then we get a biphasic curve in which you can clearly see the fast phase and the slower phase of cyclic AMP, showing that the receptor phosphorylation as well as arrestin, they can heavily influence cyclic AMP already in the first steps. So this is now what the picture we have, a preliminary picture of how the adenosine A1 gets regulated. So you have the adenosine, which binds the receptor. It activates the GI protein, which brings down cyclic AMP. The adenosine A1 then also gets phosphorylated by GRK2 and 6. And the phosphorylated receptor, along with the PIP2 binding of arrestin, gets then prepared to go into caveolae pits, but since the interaction is too transient, too weak, the arrestin basically just slides away horizontally and remains as clusters, whereas the receptor then internalizes into early endosomes. So this is at least for the A1. The stories for A2 and A2B are quite separate, but this is where I will end for this part of the presentation. I would like to thank, of course, Carsten, Jenny, and Edda, as well as the whole team of AG Hoffman. And the breadth data which you saw with arrestin was made by Alina, a student in our lab. And the cyclic AMP data was made by Caroline, a technician in our lab. And of course, I would like to thank IZKF for their funding, and of course, all of you for your attention. And if you have any questions, feel free to ask.
Thank you so much, Dr. Godbole. We really appreciate your talk. I think we have time for one quick question, if anybody has one. There wasn't, doesn't look like there's one in the chat or anything, but I'm going to ask a quick question because this is a personal interest of mine. Does this receptor signal from intracellular compartments?
That would be it would be the next step to look after this cyclic AMP thing. There are some of my preliminary data which show that there is mini GI recruitment to an internalized receptor, but it's a very shaky. But there is data that you can see mini GI on the endosomes, but mini GI also inhibit internalization, so it's a bit of a problematic thing. Yeah.
Makes sense. Thank you.
Thank you.
Thank you for this. I'm sorry. Thank you for this talk, and also for answering this question. And then I would ask you to stop the screen sharing so that our next speaker, who's Joy, can take over and start presenting her slides.
I think I already stopped it, huh?
No. Thanks. So Joy, feel free to start your presentation and also start your talk whenever you're ready.
All right, you can hear me all right?
Yeah, thank you.
Minimize. I always forget how to minimize this top row here.
I think you just need to not touch it with your mouse and then it goes away.
Okay.
We don't see it, so for us, it doesn't matter.
Okay, great. All right. Hi, thanks everyone for being here. And thanks Judith, Ian, Katrina, and Ida for organizing and hosting these talks. My name is Joy Lee. I'm a recent graduate and currently a temporary postdoc in Mark von Zastro's lab at UCSF. And today I'm going to talk about my recently submitted work currently in review. It takes the μ-Opioid receptor endocytosis as a cell biological function and dives into the agonist selectivity of that function. So the main players of the stories are the receptor, one of the subtypes of the kinase is GRK2, and these three agonists, DAMGO, a full agonist, peptide agonist that is a analog of the enkephalin, endogenous enkephalin peptide, morphine, a partial agonist derived from poppy plants, and PZM21, a partial agonist that is designed specifically for its bias towards G protein signaling. In the interest of time, I'm going to skip this. So this is no news to you. Agonist selectivity of the μ-Opioid receptors internalization. But to touch base with what's known and to kind of lay the ground, define our agonist selectivity, here I labeled cell surface receptors with a fluorescent antibody and compared agonists treated with untreated populations. And here you can see, especially at the higher concentrations, these three agonists really affect different levels of internalization. And I used the saturating concentration of 10 micromolars to plot these bar graphs to show you that they really have a widely spread or wide range of degrees of internalization, with DAMGO strongly internalizing the mu receptor and PZM21 hardly internalizing at all. And so this is how we define agonist selective internalization. We also know that this agonist selective internalization is dependent on GRK2/3. And here with GRK knockout cells generated by Asuka Inoue, Asuka is very popular today. As expected, internalization is lost even in the presence of DAMGO. However, this is rescued with overexpressing just one GRK, in this case GRK2. And not only that, but it produced a very similar dose response. And again, when I plot the saturating concentration, you see a wide range of internalization levels. So how does this single GRK distinguish affect this agonist selective internalization function? So the simplest thinking is that GRK2 is recognizing differentially activated receptor states and just interact with receptor depending on the agonist bound. I tested this idea with the split luciferase nanobit assay, putting small bit on GRK2 and large bit on the receptor. And from this time course, you can see the effect is very quick. And this is true for both the HEC293 cells with their endogenous GRKs and GRK knockout cells from Oscar's lab overexpressing GRK2, with Damgo recruiting GRK2 far better than the other two agonists, somewhere around this, over fourfold above the vehicles. And again, I plot the spread of the effect, the recruitment effect here. It's interesting that PZM21 does recruit GRK2 a little bit, even though it doesn't seem to internalize the receptor. So how is this selectivity determined? And so here I took a structure function approach and tested a few structures by either deleting or mutating them. So the first, the pH domain, because it is known to mediate membrane recruitment via the G beta gamma interaction and membrane lipids such as PIP2. The kinase domain, because it is the functional domain of this protein, and the N-terminal domain, thanks to recent structure data suggesting that it forms an alpha helix and it directly interacts with the receptor. So without the pH domain, it looks like compared to the wild type GRK, the GRK2 mutant, deletion mutant is not recruited to the receptor at all, or nearly not at all. So it looks like the pH domain is absolutely essential. Whether it is contributing or how much it's contributing to the selectivity, it is kind of hard to tell from this picture. We can maybe chat about that later or in tomorrow's breakout session. A single mutation that renders the GRK2 a non-functional kinase has this interesting property of not overall recruitment to the receptor is reduced. However, it looks like the selectivity still remains. And in a similar approach or in a parallel approach, I mutated on the receptor the stance motif that these phosphorylation substrate sites that are required for agonist selective internalization. And it very similarly has these reduced but still agonist selective recruitment to a recruitment of the wild type GRK2. So this suggests to me that phosphorylation at these sites is somehow stabilizing this interaction between the receptor and GRK2. And very interestingly, the N-terminal domain deletion did not abrogate altogether the recruitment. There's still about 20% left. However, the selectivity among the three agonists is gone. So putting these mutational studies together, it seems to me that with the prerequisite of the pH domain membrane recruitment via G beta gamma, it allows then the alpha N or the N-terminal domain to recognize, to be kind of an allosteric sensor, recognize differentially activated receptors and engage differently. And once phosphorylation activity starts, that acts as a stabilizing element for the interaction. So with this selective bind, is this selective interaction sufficient for the selective internalization function? In other words, can we manipulate the selectivity of the interaction? For example, can we force GRK2 and the receptor together? And would that eliminate the selectivity of the function? I forced GRK2 and mu receptor together with two approaches. First, with the high affinity version of the split luciferase, putting the high bit on GRK2. So even when GRK2 is kind of now sticking to the mu with the large bit, there is still selectivity when it comes to the internalization function. However, the selectivity is reduced. You can really see this with PZM21 here. It's definitely internalizing the receptor somewhat, as opposed on the left with the wild type GRK with no bit on it. I also tethered the GRK2 directly, but linking it directly to the C-terminus of the mu receptor, and that produced a very similar agonist selective internalization pattern, but the selectivity is reduced. And so since we are thinking that the N-terminal domain is recognizing differentially activated receptors, could deleting the N-terminal domain in this tethered mu receptor GRK2 molecule eliminate the selectivity? Well, it did eliminate the selectivity, but it also eliminated internalization, seems to me like altogether. So this suggests the N-terminal domain is not just recognizing different differentially activated receptors, but it is required for kinase function. So this makes sense in light of the recent structural data proposing that an N-terminal domain forms a helix as it engages with the receptor, and in that process kind of pulls the large and small lobes of the kinase domain into a closed or active conformation. So putting it all together, these data suggest that the GRK2 pH domain is essential for its plasma membrane engagement, and the N-terminal domain is an allosteric sensor that is required for agonist selectivity of this internalization function. And phosphorylation activity stabilizes this interaction. And so from a cell biological standpoint, we could kind of see these different elements as sort of a multiple checkpoint of the overall system, where the pH domain is kind of sensing, oh, there are some activated G protein activity going on, and the N-helical domain is recognizing, well, here's a fully activated receptor, and then this readies the activity of multi-site phosphorylation and follow-on arrestin engagement. With that, I'll thank my lab, the Van Zastra lab, as well as Ashish Manglik and Manglik Lab for mentoring me and collaborating on this project. I thank Aska for these GRK knockout cells, and Miriam, Ruth, and Brian for their advice and collaboration. I'm happy to take questions.
Thank you very much. That was an excellent talk. Do we have any questions for Joy? There aren't any at the moment. I'll go ahead and ask one. I'm not personally super familiar with the GRK structural arrangement and everything. To what extent do they how much do these domains vary among the other different types of GRKs?
Yeah, the GRK2 and 3 are in their own subfamily. They different from, say, GRK1, which is in the visual system, and 5 and 6. They don't have a region that kind of sticks them to the membrane. Like GRK5 has an aliphatic region. So the pH domain, also the other GRKs don't have the pH domain.
Okay,
so
thank you.
Thanks.
And Ben, did you have a question?
Yeah, I did. That was really, really great talk. I was just curious if you think that the differences you see between the different agonists is entirely efficacy-based, or if you looked at multiple full agonists, you would see variation across it based on the structure of the agonist.
Oh, that is an excellent question. I haven't tested a panel of full agonists. I did test from Brian Schoiket's lab. They had some new, actually Chase, who is now at Columbia. He through docking campaign designed a series of compounds there are among them, there are some nearly full agonists, and I do see kind of an array of responses. But no, for this particular question, we didn't follow up on those. Thanks, Ben.
Well,
And then
then sorry, you go.
Looks like maybe one more quick question from Natalia in the chat. Could the different selectivity of the ligands for internalization be due to a different conformation of the receptor in the intracellular region where it interacts with GRKs?
Yeah, I think a lot of labs are kind of thinking that way, including us. The structure data is precious, hard to come by. So that's currently with these signaling with these cell-based assays. That's how we're thinking about it. So more mutational study, more single mutations could help answer that question.
Well, then thank you for answering the questions and also for the very engaging talk. So we've heard 2 talks that featured GRKs. We're going to continue in this pattern and have a second talk featuring the μ-Opioid receptor now, which is Ben's. So you're already sharing the screen. That's perfect. And then
Is everyone seeing the full slides, not the
yes,
notes? Perfect.
it's
Perfect.
perfect. The stage is yours.
All right. Thank you so much. I guess since we have a diverse crowd, good morning, good afternoon, good evening for everyone. I'm Ben Clements. I'm a postdoc here at the University of Michigan, and I'm really honored to share our recent work looking at positive allosteric modulators of the μ-Opioid receptor and looking at how we can use different pharmacological tools to interrogate that mechanism of action. So we're particularly interested in novel therapies for chronic pain, which is a widespread issue across the world and is the most common cause of long-term disability. Huge economic costs, huge social costs, and really in terms of pharmacotherapy, as things begin to fail, the only option becomes really opioid treatment. So you get opioids, if those don't work or you become tolerant, you really have higher doses or stronger opioids become the only tool outside of surgical treatments. So we're curious, what can we do to utilize these highly efficacious opioid pathways? Because opioids do work, but limiting the negative side effects that we all know, like abuse liability, reward, and overdose risk. So we're particularly interested in developing positive allosteric modulators to act on an orthosteric site to enhance the receptor activity in the presence of an agonist, mainly because there's been some evidence suggests that we can improve the potency for specific physiological effects, ideally finding sort of a magic bullet where we can enhance the pain relieving properties without enhancing things like reward and respiratory depression. So there's been a series of PAMs developed for many GPCRs, small molecule PAMs for cannabinoid receptors, beta-2 adrenergic receptors, but we're interested in the μ-Opioid receptor, that main site of action for clinically used opioids. So 2 main classes we've looked at. The examples are going to be BMS-122 on the left here, and BMS-187, where 122 has action only at the μ-Opioid receptor, whereas BMS-187 has action at mu, delta, and kappa. So both of these compounds will in vitro enhance opioid activity. So again, as Joy introduced, we have the prototypical mu agonist DAMGO, a synthetic peptide, a concentration response curve as measured by beta arrestin recruitment. With the addition of 10 micromolar of either BMS-122 or 187, we have a left shift in that concentration response, that enhancement of DAMGO's potency. However, as you remember previously, those structures are very different, but they're doing the same thing. So we want to figure out how these compounds are affecting the receptor and how we can potentially manipulate that from a drug development standpoint. Really, the only mechanism that's been proposed comes from the Shimada group at Riken in Japan, where they used cryo-EM and NMR to find a potential binding site of BMS-122 on the outside of the μ-Opioid receptor within the plasma membrane, interacting with TM3 and TM4. So they proposed that this causes the dry residue and a tyrosine on the transmembrane 5, to interact with TM6 and lock that receptor in a more active conformation. Now, this is the proposed mechanism for BMS-122. We don't really have one for BMS-187. So what we're interested in, okay, can we use the tools that we have, different pharmacological tools, to interrogate how BMS-187's action could relate to BMS-122? And that's really the big goal of this project, is without doing these really, really intense, thorough mechanistic studies, can we start to explore whether or not these compounds might share actions at the level of the receptor? And then as we investigate that action, we want to see how different opioid antagonists, like naloxone and things like that, could interact with that allosteric response. So one tool, this is one thing that I brought to my lab from my graduate work, is to look at this pharmacologically using a method called isobolographic analysis. Essentially, this is just a straightforward statistical test. So you have different compound potencies. You do generate a, let me get a laser pointer so it looks nicer. There we go. You have an EC50 of a drug A and an EC50 of a drug B. And if these drugs are essentially acting at the same drug at the level of the receptor, so the receptor is not distinguishing them as different drugs, they will fall along this theoretical line of additivity where every single dose ratio is going to fall along this line. If the receptor is treating these drugs as different, so they're binding to different spots, they're doing different things, they're either going to help each other, which means that it will look synergistic and fall here on the plot, or they will fight each other, in which case it will look subadditive out here on the plot. And essentially, that's just a quick and easy way to look at, well, would these things potentially be doing different things at the level of the receptor? So to perform this assessment, we do a concentration response curve of BMS-187 and BMS-122 in the presence of an EC20 of DAMGO. So what we're measuring is how these compounds can enhance the beta-arrestin recruitment of that DAMGO, because these compounds in this assay do not do much on their own, at a 1-to-1, and then we do a concentration response curve at a 1-to-1 dose ratio. So half 187, half 122. It falls right in the middle, which is what we would expect for an additive response, but we want to actually determine that statistically as opposed to just eyeballing it. So what we do is we have our EC50 of BMS-122 and 187, as well as a theoretical additive point. So at a 1-to-1 dose ratio, if these drugs are treated exactly the same as the receptor, then this is where we would expect the actual EC50 we determined experimentally to fall. And as we show, it is not different than that theoretical point, suggesting that this is an additive response, which means that at the level of the receptor, these drugs are being treated relatively similarly, and that they might actually exhibit the same mechanism of action, even though we have that diversity in structure. So that was the one thing that we wanted to look at with this project. Now that we have that 122 and 187 might be acting similarly, we want to see, well, how does that relate to the orthosteric pocket? And particularly, what happens if we throw antagonists on top of this? Well, we can't put an antagonist along with an EC20 of DAMGO because the antagonist will just kick off DAMGO. Orthosterics on orthosterics, they're just going to fight it with each other. But we have one perk with the PAMs that's really helpful, and that is they have some amount of alloagonism. So because these positive allosteric modulators are driving the receptor to a more active state, they will activate the receptor to a certain extent. Normally, this can't be detected. This can't be seen in vitro or in vivo. But in highly sensitive amplified assays like cyclic AMP accumulation, you can detect that alloagonism. And from that, we can use that to probe orthosteric antagonists. If we turn on the receptor only using a PAM, can we turn it off by adding in a orthosteric antagonist without having to worry about turning off the orthosteric agonist? So this work was done by an undergraduate in our group, Tommy Prince. If anyone here is on admissions committees for medical schools, I cannot recommend him highly enough. And he performed this study. We're looking at cyclic AMP accumulation with increasing concentrations of BMS-187. And then on the addition of 3 prototypical morphin antagonists, naloxone, naltrexone, and cipridine, shown here, we are able to inhibit that alloagonism. So the binding of these antagonists to the opioid receptor is able to inhibit the alloagonism by BMS-187. So suggesting that they might be shifting the receptor into a slightly less active conformation. One thing that we did think of with this is, well, maybe the PAM could potentially be interacting with the orthosteric pocket. We haven't seen any evidence, and we've done plenty of binding studies that suggest they don't, but we want to confirm that. And interestingly, when we introduced new antagonists, some peptide-based antagonists, CTOP and CTAP, we saw no inhibition of this alloagonism, suggesting that whatever changes that BMS-187 and possibly BMS-122 by extension are inducing to the μ-Opioid receptor, they can only be inhibited by these morphin-based antagonists, whereas peptides are inducing a slightly different change to the receptor that is not able to inhibit this alloagonism. So again, wrapping things up, two kind of disparate ideas, but we are able to show that BMS-187 and BMS-122 may produce allostery via a shared mechanism of action, even though they're very structurally distinct. This alloagonism by BMS-187 can be inhibited by morphin-based antagonists, but not peptide antagonists. And then finally, these data provide evidence not only for this conserved mechanism of allostery, but they demonstrate a mechanism for probe dependence, which is characteristic of allosteric modulators, as well as a relationship between the orthosteric pocket and potential allosteric pockets. All right, with that, I would like to thank my advisor, John Trainer, as well as research faculty in our group, Jess Anand, who's fantastic, as well as the research generated by Tommy and the rest of the group. And I'm happy to take any questions.
Thank you so much, Ben. Great talk. Do we have any, Judith, did you have a question? Judith, thank you. I think you're muted.
Oh, sorry, I'm so used to just mute myself via the headphones. Sorry. If there's none in the chat right now, but there is one. Maybe we should do that one first.
Okay. So my Alberts asks, for longer term study, is a disparity of conservation between signaling allostery and structural allostery something you are considering?
Ooh, that is something I haven't particularly thought of. I've been primarily interested in the activity-based responses, not looking at changes in structure. I would imagine that pretty much everything we see that the similarity between signaling responses would relate to changes in structure. But that's something that could be different and something that we haven't really thought too much about because we're much more of like looking at the activity as related to bringing things to an in vivo drug development standpoint. It's tricky because these compounds are really hard to get on like a crystal or look at a cryo-EM. But that's something I hadn't really thought about, that there might be a distinction between the signaling allostery and the structural allostery. So thank you for that.
Michael, did you have a question?
I have one though.
That's right.
Yes, Judith.
If I may. So on the slide where you showed what happens, where the curve is shifted by the BMS compounds, it looks like the shifted curve, like, correct me if I'm wrong, but I see a bit of a biphasic curve there. Is there anything in the mechanism that's been proposed for the one compound that would fit with a biphasic curve when it's shifted?
So in the sort of earlier where it's just the damn-go concentration
Yes.
response and it left shifts for both of them, we have not seen a biphasic. We've characterized these pretty thoroughly with several different agonists. That would
Maybe
be
I'm
interesting if we
imagining
saw
it.
I mean, it's certainly possible that there are two binding sites, in which case maybe it's also one thing we thought about with regards to this sort of additive response, where if they are doing different things from different sites of the receptor, but they're close enough that we just can't detect that difference, that's certainly possible, but not something that we've seen with enough resolution to really investigate them more thoroughly. But that's interesting.
And it looks
Maybe
like
I'm just very sensitive to biphasicity right now.
If we saw biphasic, that'd be a really clear sign of two sites of action. But unfortunately, at least the curves that we've generated, we haven't seen anything consistent enough to really interrogate.
I think we'll do one more quick question from the chat. So
One more question in the chat. Oh, I'm sorry.
Okay. Have you examined the effects of trafficking behavior of the receptor with these PAMs, for example, endocytosis?
Yeah, we've done some preliminary evidence that these compounds may slightly enhance endocytosis, just really with regards to increasing the potency. I don't believe we see any increases in endocytosis that are greater than just increasing the potency of the compound. But it's something that we've only done a couple of surface level studies, haven't dug too deep into it. All right. Thank you so much. I really appreciate all of you for coming out, and you can probably find my email if you have any additional questions.
Approve.
Here, it looks like Judith is maybe having a little bit of trouble. I was having a little bit of trouble hearing her. So looks like our last one for the
Excuse me. May I ask a question, please? Sorry, I'm in delay. Good evening or good afternoon. Sorry. Can I ask a question? May I ask a question to Ben, please?
I think we're going to be moving on to our next presenter. There will be time tomorrow to ask additional
Okay, sorry.
questions.
Sorry, okay.
So thank you to Michael Wedemeyer from UT Health Science Center at San Antonio, who'll be talking about Norbinaltorphimine inhibition of peripheral Kappa opioid receptor antinociception by 1433 gamma. So thank you.
Thank you, Ian. Sorry for the mouthful of the title. Thank you to the organizers for inviting me to tell you about this project. This is one of my favorite projects in the lab, where we're investigating the Kappa opioid receptor, so following the theme, but now moving on to a different variant of the opioid receptors, and a long-term antagonist that led us to this interesting set of proteins called 1433. So pain. Nearly all of us have felt pain, and many of us don't like it. The previous presenter just told you that treating pain remains a major headache, and that opioid receptors and opioids are still some of the best analgesics we have. Opioids work by binding and activating G protein coupled receptors in the brain and the spinal cord. And as you know, opioids have many negative side effects, such as respiratory depression that kills over 80,000 Americans every year. And these effects are mediated by binding to those same sites in the central nervous system. And so a promising approach for the safe and effective treatment of pain is to instead target the peripheral sensory nervous system and stop that painful signal before it gets to the brain. So let's briefly take a look at these two neuronal types. Broadly speaking, the central neuron and the peripheral sensory neuron all express the inhibitory mu, delta, and kappa opioid receptors. In the central neuron, if we add an agonist, activate that inhibitory receptor, we inhibit that neuron from firing an action potential, so stopping this painful emotion. In the peripheral sensory neuron, we can likewise add an agonist, but we don't see activation of the receptor, and that neuron is not inhibited. So what's going on here? There is the receptor that is expressed, but something unique to the system is keeping those receptors in this quiescent state, a state we call functional competence. Luckily, we found that you can induce functional competence by adding inflammatory mediators. And under the state of inflammation, then the opioid agonist can activate the receptor and inhibit that neuron from firing, stopping this painful signal. So what does this tell us? First, opioids can indeed inhibit the peripheral sensory neurons, at least in some conditions. And second, the opioid receptors in these particular cells are regulated in a unique manner that we've not seen in other cell types. So if this is indeed something, a tissue that we can target to help relieve pain without reducing respiratory depression or the other adverse effects, then it goes without saying we're interested in how does the kappa opioid receptor get regulated in these cell types. We wanted to understand that, but we weren't sure where to look. So instead, we looked where the light was already shining, because we knew some antagonists that could lead to long-term inactivation or inhibition of the kappa opioid receptor. Here's the kappa opioid receptor, which we can activate with the agonist U50. And this leads to expected responses such as decreased cyclic AMP and antinociception, or relief of pain. The antagonist we're going to be talking about is Norbinaltorphimine, or NorBNI. So this is the prototypical antagonist of the receptor, It can sit in the pocket. It can prevent other agonists from binding, but it has this special property where a single injection of Norbinaltorphimine can inhibit core-mediated antinociception for up to 3 weeks in vivo. So this exceedingly long duration is not through irreversible binding, as you might expect. Instead, Michael Brukas and Charlie Chavkin's lab found that it's actually through activation of the Kappa receptor, biased signaling, where it activates C-Jun N-terminal kinase, or JNK. This JNK activation is required for the long-term effects of Norbinaltorphimine, and if you block JNK, Norbinaltorphimine is just a competitive antagonist. We have found that this happens in our cell type as well, the peripheral sensory neurons, and these neurons, downstream of that JNK activation, increase protein synthesis. And likewise, that protein synthesis is required for the long-term effect of Norbinaltorphimine. And so this led us to this question: what is the protein that's being upregulated by Norbinaltorphimine that's the critical mediator for the long-lasting inhibition of the Kappa receptor system? Well, I'll skip ahead quite a bit and talk to you about 1433 gamma. This protein was the most upregulated in our cell type, which are dorsal root ganglion cells, that we harvested from the rat. We added Norbinaltorphimine and found that 1433 gamma was upregulated the highest amount. So we thought perhaps this protein was involved in regulating the Kappa receptor. And there was one paper that described 1433 zeta, another family member, and that it was found in a pull down of the Kappa receptor. So we're going to test if this protein is indeed the critical mediator of this long-lasting inhibition. And to do so, we're going to use siRNA to knock down 1433 gamma. We do that on day 0 and day 3, where we also add Norbinaltorphimine or vehicle directly into the hind paw of the rat. And 2 days later, we're going to check for antinociception in the rat hind paw using this Hargreaves test, where we apply a thermal stimulus to the rat hind paw, and we measure the amount of time it takes for them to withdraw their paw. Notably, we're waiting 2 days after Norbinaltorphimine treatment, so we're not looking at the acute antagonism. We're just looking at the system that has been changed after exposure to Norbinaltorphimine. The data is shown here with paw withdrawal latency area under the curve on the y-axis. So this is the time it takes for the animal to withdraw its paw as a change from baseline over a 20-minute time course. You can see a lower number represents a more painful state or a more sensitive state as we add PGE2, which is an inflammatory mediator. This allodinic effect is reversed when we add U50-488, which is this opioid agonist. And we can see that treatment of Norbinaltorphimine 2 days prior completely abolishes U50's ability to provide this pain relief. And if we knock down 1433 gamma, Norbinaltorphimine is no longer effective at abolishing U50's antinociceptive ability. So what could we conclude from this data? It seemed that 1433 gamma was necessary for Norbinaltorphime's long-term antagonism, at least in the rat hind paw, for core-mediated antinociception. And a powerful approach to using these peripheral sensory neurons is we can actually harvest them out, the dorsal root ganglion, and measure core activation in those very same neurons that mediate the behavior I just showed you. So we do so, and we grow them in a dish and look at cyclic AMP accumulation with a similar siRNA 1433 gamma knockdown. And here's the data shown here. This is the amount of cyclic AMP on the y-axis after stimulated with PGE2, that inflammatory mediator. And this is all after using U50 as a core agonist. So we see about a 50% reduction in cyclic AMP with U50. And this is true with or without knockdown of 1433 gamma. When we add Norbinaltorphimine, we see again U50's effect is abolished. There's no reduction in cyclic AMP. But if we knock down 1433 gamma prior to Norbinaltorphimine treatment, again, Norbinaltorphimine is ineffective. It does not have this long-lasting antagonism. So just like in the rat, it seems that 1433 gamma was necessary for the signaling effect caused by Norbinaltorphimine. So to summarize this portion, Norbinaltorphimine is in fact a biased agonist at the Kappa receptor, leading to C-Jun N-terminal kinase phosphorylation and protein synthesis of several proteins, including 1433 gamma. And this leads to long-term inhibition of this pathway, where U50 binds the Kappa receptor and leads to antinociception. Ongoing work in our lab is really focusing on this dotted line here. How is 1433 gamma interacting with the Kappa receptor and causing this long-lasting effect? And is this relevant to any other GPCRs? But before I get there, I haven't even told you what 14-3-3 proteins are. They were discovered in the 60s as abundant brain proteins, making up 1% of soluble brain protein. And they're a family of 7 highly conserved regulatory scaffolds. They're found in all eukaryotes, and they function by binding proteins via these threonine binding sites and holding them together or holding them apart to influence signaling. They have no known intrinsic activity, but they can bind hundreds of targets in all cellular compartments in diverse cellular processes. And if I just explained what this was without telling you, you might expect I was explaining what beta arrestins are, another important scaffolding protein that mediate and regulate G protein coupled receptors. But in fact, we're talking about 14-3-3s and 14-3-3 gamma. In the data I've shown you today, 14-3-3 gamma expression reduced or inhibited cyclic AMP signaling and antinociception through the Kappa receptor. But what I have not had time to show you is that not all signaling is reduced, and this is not a global reduction, as kinase signaling or ERK phosphorylation was maintained. And I've also not shown you that this seems specific to the Kappa opioid receptor, as the other mu and delta opioid receptors or a serotonin receptor were not affected by either Norbinaltorphimine or 14-3-3 gamma knockdown. What I've also not had time to tell you is that this is sufficient itself. If we overexpress 14-3-3 gamma in a CHO cell, we see the same effects as if we use Norbinaltorphimine. So we abolish U50 ability to reduce cyclic AMP. And these two proteins co-immunoprecipitate, indicating there might be a direct interaction or they might be part of a larger complex. And we know what 14-3-3 proteins like to bind since there are so many targets. And looking at these 14-3-3 recognition motifs, we could identify 2 in the C-terminal tail and 1 in the intracellular loops of the Kappa receptor. So these would be cytozolically exposed for direct interaction. And if we apply the same search to the wider GPCR family, we can see that I'm jumping ahead of myself. Over 90% of GPCRs have a 14-3-3 binding domain. And so we've been using some computational tools such as AlphaFold here to get an idea of how these two proteins might be interacting, either via the intracellular vestibule or the C-terminal domain that may or may not be phosphorylated. So with that, I'd like to thank Bill Clark and Kelly Berg Lab at UT Health San Antonio in Texas, United States of America, as well as my training and my funding sources, and I'd be happy to answer any questions.
Thanks so much, Michael. I really appreciate getting a chance to see your work. Do we have any questions for Michael, either from the chat or from people who would like to ask them out loud? We don't have any questions. I'm going to briefly ask, why do you think it's gamma specifically? Because I've worked with 14-3-3 proteins a little bit too, and a lot of the time they seem to interconvert, and they'll allow, you can have different heterodimers and they'll substitute for each other. So why do you think gamma specifically?
So our work has been focusing on gamma because that's what we found was upregulated in Norbinaltorphimine. We haven't looked at other 14-3-3s if they can do a similar effect or not. They might also be heterodimerizing. The 14-3-3s generally like to form these dimers where each clamshell can bind a separate protein. So it's likely that there might be others involved, but that's not something we've looked at yet. As far as why Norbinaltorphimine increases, it seems 14-3-3 gamma specifically, that's something we're not sure. And it's not clear the pathway that's involved to induce this expression. Not all junk activation leads to 14-3-3 gamma expression. And even through the Kappa receptor, if we use a different agonist that also activates junk, we don't see these long-lasting effects through 14-3-3 gamma. So there's something unique about the pathway that it uses, how it activates junk, or possibly what junk activates after that, that triggers this one particular protein.
Makes sense. Thank you. And it looks like Sylvia from the chat is asking a technical question. How did you measure the cyclic AMP in cultured neurons?
This was a radioimmunoassay. So just doing a long time course after we culture these neurons, looking at a radioactive antibodies.
Thank you very much.
Yes.
Thank you.
Thank you also from my side. It wouldn't be a successful online conference without some technical issues. This concludes this very nice second session of our symposium about GPCR